Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore a fascinating and life-saving pathway known as glycogenolysis. If you have ever wondered how your body keeps you energized between meals, or how athletes find that sudden burst of explosive energy during a sprint, you are in the right place. Let us dive in and uncover the biochemical secrets of energy storage and retrieval. To understand what this process is, let us start with a simple analogy. Imagine your body is a smartphone, and glucose is the battery power it needs to run all of its applications. When you eat a big meal, your body plugs into the wall and charges up. But it also stores extra power in a heavy-duty portable power bank. In your body, this power bank is a giant branching molecule called glycogen, stored mostly in your liver and skeletal muscles. When you are fasting or running a marathon, you cannot just plug into the wall. Instead, your body taps into that portable power bank, breaking off pieces of stored energy one by one. This is glycogenolysis. Now that we have our analogy, let us look at the formal biochemical definition. Glycogenolysis is the specific biochemical pathway through which glycogen is degraded into individual glucose molecules to meet the immediate energy demands of the body. It is very important to understand that this is not simply the reverse of glycogen synthesis. Instead, it is a distinct, highly regulated catabolic process that involves its own specific set of enzymes. By using a dedicated pathway for breakdown, the cell can tightly control when it stores energy and when it releases it, preventing wasteful cycles and ensuring energy is available exactly when it is needed. Diving deeper into this distinct pathway, let us examine how the breakdown actually happens. The primary enzyme driving this process is called glycogen phosphorylase. Its job is to cleave the alpha-1,4 glycosidic bonds at the non-reducing ends of the glycogen branches. But here is the brilliant part. Instead of using water to break the bond, which is common in biology, this enzyme uses inorganic phosphate. This strategic, energy-saving process is known as phosphorylasis. By using phosphate, the cleavage directly produces glucose 1-phosphate. This molecule can then be easily converted into glucose 6-phosphate and fed directly into energy-producing pathways like glycolysis. Understanding this mechanism leads us to ask, why does this specific process matter so much to our daily survival? Without glycogenolysis, your blood sugar levels would plummet dangerously low just a few hours after eating, which could lead to confusion, fainting, or even a coma. Different organs use this process differently. The liver acts as a generous donor, performing this process to release free glucose into the bloodstream to keep the brain and red blood cells functioning perfectly. Meanwhile, your skeletal muscles use it selfishly, breaking down glycogen only to fuel their own rapid contractions during intense physical activities. Let us translate this biological mechanism into a formal chemical equation. On screen, we see the overall reaction catalyzed by glycogen phosphorylase. Conceptually, this shows a large glycogen polymer reacting with free-floating inorganic phosphate. Let us break down the symbols. The term G sub N represents the large polymer containing N number of glucose units. The symbol P sub I stands for inorganic phosphate, the molecule used to attack the chemical bond. On the right side, G sub N-1 shows that the main chain is now exactly 1 unit shorter. Finally, we have U, representing the single glucose 1-phosphate molecule broken off, now carrying a phosphate group. If cellular phosphate levels increase, the reaction is driven forward to produce more energy. Before we move on, let us see this with a concrete example, comparing energy costs. Suppose a muscle cell needs to process 1 molecule of glucose through glycolysis. We want to compare the initial ATP investment if the cell uses free blood glucose versus internal glycogen. Free glucose entering the cell must be phosphorylated by the enzyme hexokinase, which costs exactly 1 molecule of ATP. However, glucose released from glycogenolysis comes off already phosphorylated by inorganic phosphate, bypassing the hexokinase step entirely. Therefore, the cost for free glucose is 1 ATP, while the cost for stored glycogen is 0 ATP. This represents a brilliant biochemical shortcut. So, we have proven that using stored glycogen saves the cell 1 initial ATP. This brings us to calculating the overall energetic advantage for the cell. Let us set up the derivation for the net energy yield. We know that the cell effectively saves 1 ATP per glucose molecule when it uses its stored glycogen instead of pulling free glucose from the blood. In standard glycolysis, starting from free glucose, the pathway yields a net total of 2 ATP molecules. Because of the 1 ATP saved during the preparatory phase, the net yield for glycogen must be recalculated. Let us look at how this single saved molecule impacts the final energy budget. Continuing our derivation, let us find the final result of this biochemical shortcut. By adding the 1 saved ATP to the standard net yield of 2 ATP, we see that glycolysis, starting from glycogen, yields a net total of 3 ATP. To find the efficiency increase, we compare this new yield of 3 ATP to the standard yield of 2 ATP. This represents a massive 50% increase in net energy yield for the cell. This mathematical proof explains exactly why our muscles rely so heavily on glycogen during intense exercise. When you are sprinting, every single ATP molecule counts for survival, and this pathway delivers maximum efficiency. Now that we understand the energetic benefits, we must look at the physical limitations of this process. The enzyme glycogen phosphorylase is quite bulky. Because of its size, it cannot physically get too close to the branch points of the glycogen molecule, which are connected by alpha-1,6 linkages. The strict biological rule is that this enzyme stops cleaving when it reaches exactly 4 glucose residues away from a branch point. At this exact point, the remaining branched structure is officially called a limit dextrin. The cell must then call in a completely different protein, known as the debranching enzyme, to clear the way. This structural limitation gives us a specific mathematical formula to calculate how much glucose can be harvested from a single branch. Conceptually, this formula simply subtracts the unreachable residues from the total branch length. Let us define the variables. The term Y represents the total yield of glucose 1-phosphate molecules successfully released into the cell. The variable N sub T is the total number of glucose residues initially present on that specific outer branch. Finally, N sub L represents the steric limit of the enzyme, which is a constant value of 4 residues in human biochemistry. If the branch is longer, N sub T increases, and the yield increases proportionally. Now that we have our yield formula, let us apply it to a specific biological scenario. Imagine a specific outer branch of a glycogen molecule stored in a liver cell contains exactly 12 glucose residues. We need to determine how many molecules of glucose 1-phosphate will be released before the primary enzyme halts. We identify our total residues as 12, and we know our enzyme's steric limit is a constant 4. Substituting these values into our equation, we subtract 4 from 12. This gives us a final yield of exactly 8 molecules. These 8 molecules are now ready to be converted and sent into glycolysis for immediate energy production. To bring all of these concepts together, let us look at a visual representation of the cleavage mechanism. On the screen, you can see a branched glycogen molecule made up of green hexagons, representing individual glucose units. Notice the bulky enzyme, glycogen phosphorylase, actively attacking the terminal end of the chain. It uses an inorganic phosphate molecule to break the bond, releasing a single glucose 1-phosphate molecule, shown detaching on the right. Most importantly, look at the remaining branch. There are exactly 4 green hexagons left before the branch point. This illustrates the limit dextrin we calculated earlier, showing exactly where the enzyme physically gets stuck and must stop. We have covered a lot of ground today, so let us summarize the key takeaways. First, glycogenolysis is the essential catabolic process that breaks down stored glycogen into usable glucose 1-phosphate using the enzyme glycogen phosphorylase. Second, by utilizing phosphorylysis instead of ATP, the cell gains a massive 50% increase in net energy yield, which is crucial for intense muscle activity. Finally, we learned that this enzyme has a strict steric limit, stopping 4 residues short of a branch point and leaving a limit dextrin. You now have a complete understanding of how our bodies elegantly manage energy retrieval.